Alrighty, so we're gonna go over a week 6 lecture quiz review, guys. That's gonna cover muscles and like cardiovascular function. Okay, so let's talk about chapter 12. It went over muscles, right? So remember, there's gonna be the 4 types of tissues in the body, right? So the 4 types of tissues in the body is like epithelial tissue, connective tissue, muscle tissue, and nervous tissue. However, there's gonna be 3 types of muscle tissue, right? So you're going to find skeletal muscle tissue, right? These are going to be fibers that are going to be large, they're going to have a lot of nuclei in them, and they're going to be striped, right? Because their sarcomeres are going to be highly organized, right? So that's skeletal muscle. And these skeletal muscle, right, they're going to be able to pull on bone to produce movement, right? And so that's under our voluntary conscious control, right? Because we have like a motor neuron that's going to innervate that muscle and provide movement, basically, for a contraction. We also have cardiac muscle. Cardiac muscle is going to be striated too, just similar to skeletal muscle, because they are going to have these sarcomeres. They're not going to be as highly organized, but they are going to have sarcomeres, right? But they're going to be smaller fibers compared to skeletal muscles, and they're going to be branched. They're going to have one nuclei, right? So one nucleus. But specifically about cardiac muscle, number one, you're going to only find it in the heart, right? But these cells are going to be joined in series by these junctions called these intercalated discs, right? So it's a combination of these desmosomes holding these cells tightly together, and along with these gap junctions to allow for ion flow from one cell to the next. Really, so this muscle cell kind of acts as one whole cell, really. That's what the function of those intercalated discs are. And then we have these smooth muscles, and these smooth muscles, they're going to be small. They're not going to be striated right under a microscope, as you can clearly see in the image, right? But you're going to find these lining your internal organs, like your blood vessels, your digestive tract, things like that, okay? And so I mentioned earlier, skeletal muscles you have conscious control over. Cardiac and smooth muscles, you don't have conscious control over them, right? Now, remember that the sarcomere length will change during a contraction. So understanding that the sliding filament theory, right, is where actin slides toward the center, right, of the sarcomere. This is going to be during a contraction. So we have an increased overlapping of the actin and the myosin, right, the thin and the thick filaments, because they're going to pass, they're going to slide past each other. And so this purple right here, that's thick. This red right here, that's thin, right? So again, sliding filament theory, that actin is going to move inward, right? Sorry. The actin is going to move inward. And so these Z discs, right, they'll move closer together, okay? But that A band will remain the same length because it's just the I band that's getting smaller, okay? Again, because the Z discs are moving closer together, increasing the overlap of the thin and the thick filaments, okay? And so ATP is going to be needed, right? And so whenever we're able to create that cross bridge between the thick and the thin filament, right? So myosin and actin, right? They have this cross bridge formation. Is basically actin is bound to the myosin, okay? In order to release, in order to get that myosin released from the actin, we need ATP, right? So it's the power of ATP. When ATP binds to myosin, it loosens up that, excuse me, it loosens up that connection, right, between the myosin and the actin. And so now we can detach, right? These two molecules can detach, and the myosin can then reattach to another portion of the actin, thus providing that power stroke, basically, okay? But at the end of the power stroke, right, as you can see right here, like how that power stroke is happening, at the end of it, myosin is going to release ATP, and that kind of like helps to recock the head of the myosin. Now, in order to get that even happening, because so that's a contraction, it's the acetylcholine, right, at that motor neuron. This motor neuron, right, is going to have that acetylcholine in the synaptic terminal, stored in vesicles, of course. And so it's the release of acetylcholine from this synaptic terminal into the synaptic cleft area that's going to initiate a muscle contraction, right? So remember that acetylcholine will initiate these muscle contractions, right? And so as we can see, when acetylcholine binds, It must be 2 acetylcholine that will bind, right? It allows sodium to enter. Of course, that depolarizes the cell, and that depolarization, that sodium reaching into or getting into across that membrane, right? That's the action potential. And so we also have these T tubules in these muscle cells, and they allow for that action potential to reach down deeper into the muscle, right? And so as we can see here, that action potential is going to reach this dihydropyridine receptor right here. And this dihydropyridine receptor is a voltage-gated channel. So it doesn't necessarily open, but it will change shape, right? And so once that action potential reaches that DHP receptor, it's going to change shape. And then notice how it's physically connected to this ryanodine receptor. And so when this DHP changes shape, it opens up that ryanodine receptor. And that ryanodine receptor is a mechanically gated channel. So it's literally being pulled open from its connection with that DHP receptor. And once that ryanodine receptor is open, as you can see within this sarcoplasmic reticulum, right, calcium will then get out into that sarcoplasmic space. Okay? And so once calcium gets out, right, because we're opening that ryanodine receptor, calcium gets out, calcium will bind to troponin. Because troponin is physically connected to tropomyosin, when calcium binds to troponin, troponin undergoes a conformational change, thus changing shape. Because it's physically connected to tropomyosin, tropomyosin moves out of the way, right? Because remember, tropomyosin was blocking those active sites, but now it's moving out of the way of the active sites, thus exposing the active sites on that actin filament. And of course, myosin wants to bind in there. Okay, and then myosin binds in there, then we have a power stroke. That's a muscle contraction, right? In order to stop this muscle contracting, we have to break down that acetylcholine in that synaptic cleft, because that was the initial stimulus, right? So we break down the acetylcholine in that synaptic cleft with enzymes such as acetylcholinesterase, right? And so acetylcholine is broken down. Those sodium channels are closed. That action potential is not going down anymore. Thus, that dihydropyridine receptor closes, right? Or not necessarily closes, but it goes back to its original conformation, and it then closes this ryanodine receptor. Now calcium is locked back up in that sarcoplasmic reticulum. And so, along with us relaxing, right, our muscles, right, we're not contracting anymore, calcium will then be reabsorbed via active transport into that sarcoplasmic reticulum, and then we'll also pump out that sodium via that sodium potassium exchange pump as well. Okay. Excuse me. These are just going to be the 3 types of muscle types that we just went over. And understand that what you see is constant with that calcium. Calcium is going to be important for all type of muscle contractions. Okay. Skeletal muscles contract the fastest, where smooth muscles are going to contract the slowest. Cardiac is going to be in between them. Okay. Again, skeletal muscles, they require acetylcholine from a motor neuron, so we have conscious control over it. However, smooth and cardiac muscles, they do not, right? So it could be the autonomic nervous system, stretch receptors, chemical signals such as hormones, things like that can stimulate their contraction. Okay. When we're looking at the integrative physiology, right, remember that these are going to be the 4 types of classifications of a neural reflex. Number 1, we can classify a neural reflex by the efferent division that controls the effector, right? So if it's a somatic or an autonomic reflex, right? That's the efferent division, right? Going toward either muscle, somatic, or gland or organ, autonomic, right? We can also classify neural reflexes based on the location where that reflex is being integrated. Is it a spinal reflex or is it a cranial reflex, right? Is it being integrated at the spinal cord of the brain? We can also classify a neural reflex based on if that reflex is innate or learned, right? Are you born with it or did you learn it through conditioning? We can also classify a neural reflex based on the number of neurons in that reflex pathway. Is it a monosynaptic reflex or a polysynaptic reflex? A monosynaptic reflex is where we just only have one synapse, right? If you have one synapse, you have 2 neurons, okay? And so this is an example of a monosynaptic reflex, such as an example of like the patellar reflex, right? This is also a somatic reflex because it's going to muscle tissue. So we can classify this as a monosynaptic somatic type of reflex, right? But again, these somatic motor reflexes, they're going to go to muscle, right? An example of a polysynaptic reflex is you're going to have many, right? So you're going to have many synapses. So we have one synapse, two synapses. So a polysynaptic reflex is two or more synapses. Remember, a synapse is between a neuron and another neuron, or a neuron and a gland, or a neuron and a muscle, etc., right? So we can see that if you have two or more synapses, we're going to have 1, 2, 3 neurons. So at least 3 neurons in this pathway. Okay? And the example of a polysynaptic reflex is a cross-extensor reflex. Okay? Remember that autonomic reflexes, so they're not going to go to muscles, right? But they're going to go to glands and organs and adipose tissue, things like that. And so these autonomic reflexes, all of these autonomic reflexes are going to be polysynaptic, okay? With at least one synapse in the central nervous system and another in an autonomic ganglion. So here's at least one in the central nervous system between that interneuron. Remember, interneurons are only situated in the central nervous system. So we can see at least one of these synapses, I'll give you an example of 2 of them right here, are in the central nervous system, and another one in an autonomic ganglion right there. Okay. And when we're looking at these autonomic reflexes, they can also be called visceral reflexes, again, because they're involving glandular tissue or organs, right? They're not targeting skeletal muscles, right? But some of these spinal reflexes, I'm sorry, some are going to be spinal reflexes with brain modulation, and some are just going to be integrated in the brain. Now, some are just integrated in the brain, and not all reflexes require input from the brain, okay? So we have like tonic activity, right? So remember these. Tonic activity just means, excuse me, you're having a constant stream of action potentials that's going to create an ongoing activity in that effective tissue. So, for example, your blood vessels are under tonic control, continuous active autonomic reflex would be an example. Then we have the integrating centers, right, for autonomic reflexes. They're going to be in the brain. Okay, they're going to include like the hypothalamus, the brainstem, and that limbic system. And those autonomic reflexes, they're going to be such as vomiting, salivating, swallowing, and sneezing. Now, sensory receptors are known as proprioceptors, right? Sensory receptors are these proprioceptors. They're going to be located in skeletal muscle, joint capsules, and ligaments, right? And so these input signals from these proprioceptors, they're going to go to the central nervous system through these sensory neurons. And so these proprioceptors, they're going to tell us about our body in space. So we're going to have proprioceptors, or these sensory receptors, right? Such as the Golgi tendon organs, the muscle spindles, and the joint receptors, right? These joint receptors, these are going to be found in the capsules and ligaments around joints in the body, okay? And they're going to be stimulated by mechanical distortion in that joint capsule. And so that sensory information from the joint receptors, they're going to go right to the cerebellum, maintaining posture, equilibrium, balance, things like that. And we also have these, what is known as alpha gamma co-activation, right? Where we have the simultaneous activation of the alpha and the gamma motor neurons. Remember that when we're looking at, these are our extrafusal fibers, right? That muscle tissue that we've always been talking about. And inside that muscle, as you can see, intercalated between the muscle, we have this muscle spindle, right? And that's going to be called intrafusal muscle fibers, right? So it's this alpha gamma activation through these muscle spindles. And so these extrafusal muscle fibers, they're basically monitored, right, by these muscle spindles to ensure that you don't overstretch them. And so the function of these muscle spindles that I'm circling right here is to monitor muscle length and prevent overstretching. So when a muscle contracts, the alpha gamma coactivation ensures that the muscle spindles remain active, right? Activation of these gamma motor neurons causes the contraction of the ends of the intrafusal fibers, right? So these gamma motor neurons will go to the spinal cord, right? And that's going to be able to synapse and give you control honestly prevention of overstretching over those extrafusal muscle fibers, right, through these alpha fibers, okay? But again, we have alpha gamma co-activation, that is simultaneous activation of the alpha and the gamma motor neurons between these muscle spindles in the muscle. And the function of this muscle spindle is to monitor the muscle length and prevent it from overstretching. We also have these Golgi tendon organs, right? They're going to respond to muscle tension, okay? And so these Golgi tendon organs, right, as you can see down here, they're going to be found in junctions of tendons and muscle fibers, right? They're going to be composed of free nerve endings that wind between the collagen fibers inside the connective tissue capsule. Excuse me. But again, they're going to respond to muscle tension developed during isometric contraction, right? And they're going to cause a relaxation reflex, okay? And so sometimes you'll find that they'll inhibit more neurons when there's an excessive force exerted on this muscle spindle. Again, responding to muscle tension, okay? Reflex movements, right? These are going to be the least complex. They're going to be integrated at the spinal cord of the brainstem. And they're going to be regulated by negative feedback. We have postural reflexes. Postural reflexes are going to help us maintain body position. They're going to be integrated in the brainstem and spinal cord, okay? They're going to require continuous sensory input from the visual and vestibular sensory systems, okay? And so we have postural reflexes, rhythmic movements, voluntary movements, right? Things like that. Most of these reflexes are going to be integrated in the spinal cord, and they're going to be regulated by, again, negative feedback, okay? Now, we did talk about Parkinson's disease, which is characterized by the loss of motor control, right? And so it's a progressive neurological disorder. It means it's going to continue to degenerate, and then symptoms we see abnormal movement, speech difficulties, cognitive changes, and it's going to be due to a loss of basal ganglia neurons that release dopamine. So those dopaminergic neurons, they're going to be lost in the basal ganglia, right? And so if those neurons are lost, we have a loss of dopamine. No cure, but we do treat it with the administration of L-Dopa, right? So dopamine can't cross the blood-brain barrier, but a precursor to dopamine, L-Dopa, can cross the blood-brain barrier. So once it's crossed the blood-brain barrier, then it gets converted to dopamine, okay? Then when we're looking at cardiovascular physiology, right? It's cardiac muscles that, these cells that can contract without innervation, right? They don't need the brain to control their contraction because they are myogenic, originating within the heart muscle itself, right? So the signal for that local contraction, right, doesn't come from the nervous system, but it does come from these specialized myocardial cells known as those autorhythmic cells. That's going to be your pacemaker cells. And these cells will induce the signal for contraction. They're going to set the rate of the heartbeat, okay? And then we have, of course, those pacemaker cells. That's going to set the heartbeat. And then we have these contractile cells. These are going to be those muscle cell fibers. Now, when we're looking at an action potential of cardiac cells, so we talked about an action potential of like nervous tissue, right, or skeletal muscle tissue. However, when we're looking at the action potential of a cardiac contractile cell, you'll see these different phases right here. So this, this is zero phase basically, right? This is the depolarization phase. That depolarization phase is where the sodium channels are open, okay? So depolarization is where sodium channels are open. And then once we get to around 20 millivolts in charge, right, those sodium channels will close. And then we're going to have these slow, funny channels, right? They're going to open. Those potassium channels are going to open, okay? And so when these potassium channels open, so we become more permeable to potassium, and we also become more permeable to calcium. And so you have potassium leaving the cell and calcium entering the cell, and that's going to give you that plateau phase right there for this action potential of the cardiac contractile cells. Okay, and so then we're going to get these calcium channels, they're going to close, and then these potassium channels, they're going to then open. And so then we have the repolarization of the cell when those potassium channels open, okay? And then we get back to our resting membrane potential, Okay, so remember these different phases of myocardial contractile cells. Okay, and again, that plateau phase is going to be this long action potential that's due to calcium inflow and potassium outflow. Okay, and so the other action potential, right, that's going to be in the cardiac autorhythmic cells, so those pacemaker cells. So we're going to find a new action potential in our SA node. And again, the SA node is your pacemaker. It controls your heart rate. Okay, and so what I really want you to look at right here is this pacemaker activity is going to be due to those funny channels, right, which are going to be permeable to both potassium and sodium. But excuse me, this pacemaker action potential is really going to be due to calcium coming in. Calcium will depolarize the cell, and potassium going out, right? So potassium will repolarize the cell. And again, this is the action potential in your SA node, okay? Now, because your SA node will fire, that information, electrical signals will travel through these internodal pathways. It'll go to your AV node, right? Things like that. And then that electricity will continue to travel down the bundle of His, or the bundle branches, to the Purkinje fibers, right? And so it's the Purkinje fibers, they're actually able to transmit electrical signals down, right, the atrioventricular bundle to the left and the right bundle branches, okay? And so, or they can express that electricity and help with those contractions of those ventricles, really. And so it's the SA node that sets the pace of the heartbeat around like 70 beats per minute, right? But say, for example, something is wrong with the SA node, your AV node can kick in, and that'll set the heart rate around like 50 beats per minute. Something's wrong with that. Then those Purkinje fibers, they can act as a pacemaker, right, under certain conditions, and they can set your heart rate to around like 25 to 40 beats per minute, okay? And it's that AV node that has that delay with the slower conduction of signals through those nodal cells, okay? So doing isovolumic ventricular contractions, both the atrioventricular valves and the semilunar valves, they're going to be closed, okay? The ventricular blood volume is not going to change, but the pressure will rise. When the ventricular pressure exceeds the arterial pressure, then those semilunar valves are going to be open and blood is going to be ejected into the arteries, okay? And so, excuse me, we're also looking at an electrocardiogram, right? And so this is going to show the summed electrical activity generated by all the cells of the heart. So it's not going to be the same as an action potential, though. But when we're looking at the waves of an ECG or EKG, right, we're going to have three typical waves, right, that we'll see. The P wave, remember that the P wave is where the atria contract. And if it's where the atria contract, this is the depolarization of the atria. So P wave is depolarization of the atria. The QRS complex is where the ventricles depolarize. And that means the ventricles are contracting, okay? And then we have the T wave. The T wave is repolarization of the ventricles when the ventricles relax. Right? And we didn't see where the atria relaxed, right? And so while the atria are repolarizing, while they're relaxing, the ventricles are depolarizing. But because the ventricles are depolarizing with so much force, it kind of outweighs that atrial repolarization. So that atrial repolarization, it's really part of the QRS complex, it's just hidden, okay? And so we have also, remember when we were talking about that plateau phase and that cardiac contractile cells action potential, when we're looking at an EKG, right, is this ST segment that is going to be that actual plateau phase. And also we have progression of the impulse through the AV nodes. They can be seen on an EKG as that PR segment, okay? It's that PR segment. Remember, systole or systole means contraction, right? If you're contracting, these ventricles are pumping out blood. Diastole or diastole, that's relaxation. That's where they're filling up with blood, right? And so also remember stroke volume. Stroke volume is simply the amount of blood pumped by one ventricle during a contraction, right? And so the stroke volume is going to be equal to the end diastolic volume minus the end systolic volume, okay? Cardiac output is just the volume of blood pumped out about a ventricle in 1 minute, okay? Remember these heart sounds, lub, dub, right? Lub is where the AV valves are closing. Dub is where the semilunar valves are closing. Okay. And so there's going to be multiple factors that influence stroke volume, right? So stroke volume is going to be proportional to the end diastolic volume, and that's just simply the volume in the ventricles at the end of ventricular relaxation, okay? And also other influences of stroke volume is force of contraction, right, in the stroke volume, and in the venous return as well, right? How much blood is being returned back to the heart. And that's it, okay?